Here. So first of all, thank you very much for the invitation to come and talk. I had been hoping to come along in person, and when Ruth first invited me, the idea was I would come along to your meeting. Clearly events kind of overtook all of that, but it's really great to be able to chat to you virtually, if not in person. So what I've done is as Ruth said, I work on basically the sort of molecular cell biology of Parkinson's. And this is what I've been doing for actually about 15 or so years now. So what I thought I'd talk about is basically where the research that I do comes from and how that's helping us to understand some of the changes that are occurring in the brain of people with Parkinson's. And more importantly, how that's helping us develop new approaches that are actually now going into clinical trials. So I'll talk a little bit about some of the drugs that are now undergoing clinical trials that have come out of genetics and our understanding of genetics. There we go. That should be up on full screen. So the sort of catch-all title I've used is using genetics to develop new drugs for Parkinson's. And actually, I think that pretty much captures what it is I'm going to be talking about today. So clearly, we've known about Parkinson's for many, many years now. For those of you who haven't seen it, this is the original description of Parkinson's by James Parkinson, noting that he didn't call it Parkinson's disease. He called it the shaking palsy. This is actually what we think is the only image of James Parkinson. Unfortunately, he died before cameras were invented. But this is an illustration from one of his pamphlets where he was giving a sermon to people at a pub about living a healthy life. So he was kind of like a general practitioner, and actually lived a fascinating life. And there's some very interesting stuff on the internet about his life and his description of it. So we've known about Parkinson's for many, many years, over 200 years now. But actually we've struggled to understand what causes it. And the way in which myself and many of the academics, the geneticists and clinicians and people with Parkinson's that I work with, the approach that we've taken to try and understand Parkinson's is actually to use genetics as a way of doing this. And the key bit of information that we use for this is actually a family tree. So this is a family tree that I've cobbled together. Here's the sort of the youngest generation down here. These are their parents, grandparents, and great-grandparents up here. And what I've done is I've used an example of a family tree that some of you may recognize. I'm not going to ask because I'm sure it will get very complicated with everybody zooming in. But this is the royal family tree. And for a variety of reasons, it's actually quite a useful one to illustrate the point of how we can use genetics to understand a lot about life, actually, and how we look, sometimes how we behave, and sometimes how likely it is we'll develop different disorders or diseases, sometimes very early in life, and sometimes much later in life. And the way in which I'd illustrate this, actually, is to highlight Prince Philip up here at the top, Prince Charles down here, Prince William, and highlight the fact that, first of all, if you look at them, you can see that their faces are quite similar. And in a way, this is something that all of you will be aware of, that people that you're directly related to look like you. Equally, there's something that you can see quite clearly is being passed down from generation to generation here, and that's male pattern baldness, which does not bode well for Prince George, unfortunately. And clearly, as those who have seen my video, this is a subject that's very, very close to my heart. So that's an example of how a trait, something that you can see in individuals, can be passed down from generation to generation. And what drives all of this is the instruction book, if you like, that is found within all our cells, the DNA, the deoxyribose nucleic acid. And you hear a lot about this in the news, in health reports, and in the popular press more generally now. This is actually what your DNA looks like. These are the human chromosomes. So almost every cell in your body has lots of these chromosomes, and essentially what they are is an instruction book. And in that instruction book, you have the instructions to make all of the machines that allow your cells to function, to allow you to digest food, to allow you to grow, to ensure that the follicles on your head keep growing throughout your life. And this is a sort of zoomed in version of that. So this is a human cell. And if you were to zoom in using a very high-powered microscope, you'd be able to see the individual chromosomes. What you wouldn't be able to see, even with the most powerful microscope, is what these look like at a molecular level. So as we go around this sort of loop, going around to the gene and to the nucleotides, this is actually beyond the resolution of any microscope. You couldn't use an electron microscope, which is the most powerful type of microscope we have, to look at these. But using a lot of very, very advanced science and some not so advanced science, actually this is what we know DNA looks like. It's this tightly coiled ribbon of information made up of individual bits of information. So if you like, it's an instruction manual with 3 billion letters in it. And those letters and the way in which they're arranged allow our cells to function and allow us to live. They're the instruction manual for our cells and for our bodies. Now, every so often, that instruction manual makes a mistake when it's being copied, and the words and the letters perhaps end up in the wrong order or are disrupted in some way. And these are what we call mutations. And one of the things that can do is it can cause disease of one type or another. The example which we understood first is actually examples like cystic fibrosis or sickle cell anemia, where an individual change, just one little change in those 3 billion bits and letters of information, one little change could cause someone to develop a disease that presents with symptoms and can cause quite serious problems. And we now know a lot about what these mutations can do. We're able to sequence a whole human genome now, actually relatively cheaply and relatively quickly, within a day, and at a cost of just under £1,000 now. Now, what has all this got to do with Parkinson's? Why is this of interest and importance in terms of Parkinson's? Well, most people who develop Parkinson's, I'm sure you will be aware of this, most people who develop Parkinson's don't have a family member or a member of their close family, a brother or sister or parent or uncle or aunt who also has Parkinson's. But a minority, a relatively small minority, probably somewhere between 5 and 10% of everybody who develops Parkinson's does have a family member also affected. And actually they develop Parkinson's because they have one of these mutations in their DNA and in their genome. And actually we began to understand this over 20 years ago now. And it's quite often you hear the term sort of paradigm shift and step change in our understanding of health. For Parkinson's, actually, and from my understanding of Parkinson's, you can identify one of those going back to 1997. And this was a study carried out by a neurologist over in America, a guy called Bob Nussbaum, who's shown over here on the left. He was working with some individuals with Parkinson's, living with Parkinson's, from a family in America who were originally from the Contersi region in Italy. And they were one of these families that had a very strong family history of Parkinson's. What you can see here in the middle of the screen is their family tree. And what's different about this family tree compared to the royal family tree is that all of the filled-in shapes, so a square is a male, a circle is a female in this family tree, each of those shapes that's filled in is an individual in the family who developed Parkinson's. And very quickly you can look at it and see that actually about half of this family developed Parkinson's during their lifetime. This is very, very rare, and actually we now know that this particular form of inherited Parkinson's is very, very rare, only a handful of families around the world. But from a genetics point of view, you would look at a family like this and say that it's very, very likely that they have a mutation, a change in their DNA, which is causing them to develop Parkinson's. And actually, this is what turned out to be the case. So Bob Nussbaum, working with geneticists and working with other clinicians and working with the individuals in the family, was able to extract DNA and sequence their genes and identified a mutation, one of those changes in the DNA, in a gene called alpha-synuclein. Now, we now know actually that alpha-synuclein is very important in Parkinson's. And it's the gene that makes the protein alpha-synuclein, which is found in sticky lumps in the brains of many people, if not all people, with Parkinson's. And so going back to 1997, actually this changed the way in which the scientists and neurologists thought about Parkinson's, because it gave an insight that there were changes in DNA that could give an insight as to what was happening. So since then, in the last 23 years, a huge amount of work has gone on working with people and families who have Parkinson's to see if there are other genes that are changed in people who have Parkinson's. I'm very fortunate to work, I'm not a geneticist myself, I'm a biochemist and cell biologist, but I'm very fortunate to work with a number of geneticists who have really changed the way in which we think about the genetics of Parkinson's. The guy down here in the right-hand corner is Andy Singleton, who is a leading researcher over in America, working for the American government at the National Institutes of Health. And he has been instrumental in changing the way in which we think about the genetics of Parkinson's. And what's striking is that actually there isn't just one or two genes which are involved in the risk of developing Parkinson's, but actually there are many. This is a list from one of the papers that he's recently published earlier this year, summarizing the genes that we know about involved in Parkinson's. So you have somewhere around 20,000 genes in your DNA. So if you were to look inside one of your cells, there's probably somewhere around 20,000 different genes across those 3 billion base pairs and 23 chromosomes. And we've now got a list of about 20 of those genes, where if you have a change in the writing, a change in the letters, actually that can cause you to develop Parkinson's. And I'll emphasize again, this is a rare cause of Parkinson's, but it's one that we can begin to understand. And actually, since 2005, I was working in the lab that Andy runs over in America. I spent 2 years working over in Washington on Parkinson's, where I first started working on Parkinson's. And we were working with one of these genes, and it's on this list, the fifth in this list, a gene called leucine-rich repeat kinase 2, or LRRK2, LRRK2. And again, this research can only come about through working with individuals affected by Parkinson's, families living with Parkinson's. And this is actually an English family, a family from the east of England. And it's the same kind of family tree. And here, each diamond represents an individual in the family. And again, the ones that are filled in are the people who develop Parkinson's. And it's very similar to that family from Italy that Bob Nussbaum was working with. About half of the people in this family developed Parkinson's, and they just didn't understand why. So they worked with colleagues of mine at University College London, working at the Institute of Neurology there, and very kindly donated samples of their DNA to be analyzed. And what turned out was that there was a single change in those 3 billion base pairs of DNA, 3 billion letters of DNA. There was one change that all of the people in this family who developed Parkinson's had, but the unaffected, so people who didn't develop Parkinson's, didn't have that change. And what that allowed us to figure out was that this change in their DNA was causing them to develop Parkinson's. So this was actually identified back in 2004. And since then, a lot of information has come out relating to this gene LRRK2. And it's one that I'm going to talk about for the rest of this talk. Now, for most genetic diseases, actually, you don't have a public face for mutations that cause this a particular disorder. But for the LRRK2 gene and for Parkinson's, actually, we do. So this is Sergey Brin. He's one of the co-founders of Google. And about 10 years ago, his mum developed Parkinson's. And it so happens that his wife at the time, a lady called Anne Wojcicki, who founded a company called 23andMe, she was and is a geneticist. And she said that given his family background, he's an Ashkenazi Jewish background, so European Jewish background. for a lot of different reasons, some of which we understand, some of which we don't, there's an increased rate of some mutations that can cause Parkinson's in Ashkenazi Jewish families. And so Amojiki said to him, you should probably get your DNA looked at, and he did. He had it sequenced, and it turns out that actually he's got one of the mutations that can cause you to develop Parkinson's. He's actually in his late 40s at the moment, but there's a fair degree of certainty that by the time he's 80, he will go on to develop Parkinson's. And I think that's actually a very good example of how genetics is changing, and the new genetics, if you like, the ability to read our DNA is changing our relationship with that instruction manual within ourselves. So he hasn't got Parkinson's at the moment. He doesn't have any of the symptoms of Parkinson's, but it is very, very likely, and we can predict with a degree of certainty that he will go on to develop it, which is actually quite astounding. Even 15 years ago, that would not have been possible. What's also striking, of course, is that because his ex-wife ran 23andMe, actually this is a mutation in a gene that you can get looked at if you so desire. So for 23andMe, they do personalized genomics. You can send off a sample of your spit, so essentially a sample of your DNA, to California, and for 125 quid, you get a readout of your DNA. So I can tell you things that you probably know about already, like your eye color, but it can also tell you about potential risk for certain disorders, heart disease, dementia. Parkinson's is one of those things that they look at. And actually you can find out if you have the mutation that Sergey Brin carries in the LRRK2 gene. Now, whether you would want to find that out is a very, very different question. But actually this is technology that is within a lot of people's reach now. And so it's something that I talk to my students at the veterinary college, and I point out to them that actually their generation is going to be the first really to be able to read their own DNA and find out what it actually means, for better or for ill. So what does all of this genetic information mean in terms of developing drugs for Parkinson's and understanding what causes Parkinson's to develop? Why is it that some brain cells die quicker than others? So this is basically what I've worked on for the last 15 years. And this is a sort of cartoon of a generic brain cell, a neuron. What you've got here is the cell body, which is where all the sort of the factories and the energy is generated. And you have a long bit that extends out called an axon. And this is what's involved in passing information from one cell to the next. And what we've learned about over the past 15 years about LRRK2, by looking at these mutations, and we can take these mutations and put them into cells that we grow in the lab. I'll mention very briefly later on, we can actually genetically engineer simple organisms so that they have those same mutations. And we work with a very simple worm called C. elegans in the lab, which allows you to actually look at what happens when you insert that mutation. You can find out what that mutation does. What we found looking in human cells that we can grow in the lab is that some of the ways in which a cell organizes itself, some of the ways in which it moves stuff around the cell and collects waste, are changed if you have a mutation in this particular gene. And what we found is that a bit of the cell called an autophagosome is disrupted. It changes its function when you have a mutation. This is a microscope image of a human cell that we've grown in the lab, and we've used a powerful microscope that uses lasers to generate images. This is what it looks like at high resolution. Actually, what you can see here is those green specks are autophagosomes. And these are very, very important bits of the cell. Very, very broadly, what they do is they collect rubbish. And this is the Reading Borough Council Waste Service, who goes around and picks up our recycling. Very broadly, this is what an autophagosome does in your cells. And they form these sort of open C-shaped accumulations within the cell. My former postdoc, Claudia Manzoni, who has actually now has her own research group at UCL, used to think that they looked a lot like Pac-Man. And actually that's not a bad way of thinking about what they do, because what they do is they go around the cell and they identify rubbish that needs collecting and being got rid of. So what I've put here is, in orange over on the right hand of the slide, are two mitochondria. Mitochondria are they're a bit like the batteries of a cell. They generate energy. They allow your cells to function. Now, like any battery, sometimes they go wrong. And when they go wrong, and I've colored them red to make it look as though they've gone a bit haywire, an autophagosome will come along, gobble them up, close around them, and then take them to another bit of the cell called the lysosome, which has lots of specialized cutting machines, enzymes, which can basically break up and degrade the broken mitochondria. And it can do this for all types of different waste within the cells. So, and you find these in all cells, by the way. So if you were to look in a liver cell or a heart cell, you'd find the same machinery, the same process going on. So what do we think this might have to do with Parkinson's? We have an idea that if you have a mutation in LRRK2, actually something about the way in which these work isn't working quite right. So what we think is happening, as with much of science, we're not sure that this is what's happening, but we have some evidence to support that this is the case, is that it might have something to do with that protein that I talked about at the start, alpha-synuclein. And we've known now for over 20 years that alpha-synuclein forms lumps in the brains of people living with Parkinson's. It's one of the key changes in the brain that we know about. And these are basically brain cells chock full of alpha-synuclein, forming great big sticky lumps. And you could sort of, almost like a sort of tangled ball of string that clogs up the cell and causes the cells to dysfunction, and we think actually causes those cells to die. So this is one of the examples where the genetics has actually helped us to understand what's going on in terms of brain cells. So we think that alpha-synuclein itself, when it forms these lumps, these knots, can actually cause cells to die. Now, normally in the brain, what would happen is that when you get protein accumulating as sticky lumps, you get an autophagosome. It would come along, it would gobble up that rubbish, close around it, and degrade it, get rid of it, recycle it. So what we think might be happening in people who have a mutation in LRRK2 is that something about that process isn't quite working. And so instead of going around and capturing these sticky lumps of protein and degrading them and getting rid of them, what you get is you get an accumulation of that protein. And there's something about that process of accumulation which causes brain cells to die. And actually, this might be going on in Sergey Brin's brain at the moment. We don't know, is the honest answer, but it's something that could be happening in his brain at the moment. So what we think is happening, and this is work that has gone on in my lab for a number of years now, is that LRRK2 is working a bit like the sort of local council office. It's working as a control center that sends out these autophagosomes, gets them where they need to be, clears up the rubbish, and then takes it away. And if you have a mutation in LRRK2, and this is a model for what we think LRRK2 looks like at a molecular level, if you have a mutation in this gene, what happens is that those messages get garbled. They don't go out to the autophagosomes properly, so the autophagosomes don't go to the right street address to pick up the rubbish, or they collect it and then put it in the wrong place. And that something about that process is causing brain cells to die. There's a lot we don't know about LRRK2. There's a lot that we need to learn about it, but most people agree, most scientists working on it agree that this is probably something along the lines of what is going wrong with LRRK2. And it might not be autophagosomes, it might be another cellular process, but it's something about that sort of command and control of how the cell works that's going wrong if you have a mutation. Now, how does this all fit into drug discovery? So it turns out, and actually this was obvious back in 2004 when the mutations were first described, that LRRK2 is a specialized type of gene, specialized type of protein called a kinase. Basically, kinases stick small molecules onto other proteins and change what they do. And that's how they exert control. That's how they guide cellular function. One of the important things to know about kinases is that they are very favorably looked upon as drug targets. If you look across to cancer research, a lot of the drugs which are now in clinical practice and are being used to treat people who have cancer, a number of these target kinases. And so the large pharmaceutical companies have for many, many years worked to understand and try to develop drugs that target kinases. So when LRRK2 came along, actually the pharmaceutical companies immediately sat up and took notice, and they started getting their medicinal chemists to develop compounds, small molecules, that could be developed into drugs. And these are examples of some of the chemical structures of drugs that large pharmaceutical companies like Black have been developing to try and modify the way in which LRRK2 works. And basically try and correct those problems that occur when you have a mutation. And actually, this has developed quite far. Over the past 15 years, this has gone from working in cells and working in mice to try and understand what these drugs do, to going through to clinical trials. And it's worth, I'm going to pause very briefly here just to briefly outline the way in which clinical trials work. And obviously, this is something which a lot of people are learning a lot about at the moment because of the vaccines going through coronavirus clinical trials. So basically, the way in which clinical trials are organized is in 3 phases. Phase 1 is a safety phase. You give someone a drug or a vaccine for that matter and see whether or not it's safe to give them. You don't want to give someone a drug or a vaccine that causes them severe harm and is worse than what you're trying to prevent them or treat them for. Phase 2 is if something gets through that initial test to see whether it's safe or not, to see whether or not it actually makes a difference to the disorder or infection that you're looking at. Then you go on to phase 3. These are the big, large-scale trials where you may have thousands of people taking a drug to see if it really does make a difference to people who have those particular set of symptoms, infection, or disorder. What's useful to remember in all of this is how likely it is these things fail. So phase 1, and this is data from actually from a few years ago now, about 6 or 7 years ago, where a team looked at all clinical trials across every type of human symptom and disease. So at that first stage, about 40% of all clinical trials fail. You get to phase 2, about 64% fail. You get to phase 3, and about 38% fail. So quite quickly you can see that most drugs that start out in phase 1 trials do not get through and pass phase 3. And it's only after something passes phase 3 that it can go into clinical practice and actually be recommended for human use for that particular set of symptoms or infection or disease. The other thing to note is the timeline here. So the first stage can take between 2 and 3 years. Phase 2 can take 2 to 3 years. Phase 3 can take 3 to 4 years on average. This can be sped up, and the current coronavirus vaccines are a very good example of this. But it gives you an idea as to exactly how long clinical trials take. Also gives you an idea as to how much they cost, by the way, which is hundreds of millions of dollars. But actually, we're beginning to go into that pipeline, that process of deciding whether or not targeting LRRK2 will make a difference. And Denali Therapeutics is a biotech company over in San Francisco who have taken one of these drugs and have started testing whether it's safe to give this to humans. And actually, they've completed a phase 1 clinical trial. They completed it last year and are now starting to give this drug to people with Parkinson's. Initially, people who have that mutation, who have that the mutation in LRRK2. But eventually what they want to do, if it's successful in people who have a genetic form of Parkinson's, they want to try this with anybody who has Parkinson's to see whether it will make a difference. And I think actually, again, this gives you an idea as to the timescale, the very frustrating timescale that it takes to go from an initial insight into what might cause Parkinson's through to a clinical trial. So those original papers back in 2004 that first described mutations in LRRK2, they were back over 16 years ago now. Over the 16 years since then, there's been a huge amount of research carried out by labs across the world, including my own. But it's only in the last couple of years that those have actually come and gone into clinical trials. And for those of you who've read the papers about Alzheimer's clinical trials, you'll know that actually a majority of those trials have ended in failure. And it's likely that many of the clinical trials that are ongoing and have started for Parkinson's will fail for lots of different reasons. But the hope is that by understanding the changes, the underlying changes that alter the way in which brain cells can work and can cause them to die in Parkinson's, we're beginning to make that leap into intervening, developing drugs and approaches. And actually, only last year, another study was started, again targeting LRRK2, using genetic engineering, using a gene therapy approach to try and modify what the LRRK2 gene does and to try and prevent it causing Parkinson's. So there's a huge amount of activity, a huge amount of work going into trying to develop drugs. But all of this comes from working with the families who have these inherited forms of Parkinson's. And my own work actually is broadened out from cell biology. We're doing lots of different things to try and understand more about the LRRK2 gene. This is the Diamond Light Source, which is the UK particle accelerator up at Harwell, which is just around the corner from Didcot, for those of you who know where Didcot is. And they do some amazing structural biology there, where they use the particles going round this great big donut. It's about 600 meters across, to give you a sense of scale. They use that beam of light to understand how molecules function at a molecular level. We're working with them to try and understand the structure of LRRK2 and try and understand more about why a mutation, a change in that gene, can cause Parkinson's. We're also working, funded by Parkinson'S UK actually, to try and understand what that can do to whole organisms. So this slightly horrid beast crawling across your screen is C. elegans. And fortunately, C. elegans is not really that big. It's about 10 millimeters across at the very most. But we actually have a PhD student, Rachel Chandler, who's down the right-hand corner of the slide, who's working with a C. elegans specialist at the University of Reading, Ava Kavai, to try and understand what happens when we genetically manipulate, when we engineer these worms to carry that same mutation that Sergey Brin has. And we're actually already learning about what that does to the worm and the changes that that has on the way in which their brain cells work. They don't have many brain cells. They have about a dozen in total, I think. So not the most advanced brain of all time. But actually that simplicity helps us. It allows us to experiment with them, to give them the same drugs that are being used in humans, to try and understand what the consequences of that are. So hopefully, I've probably rattled on a little bit longer than I'd said I would. Hopefully that gives you a bit of an insight into how understanding genetics, understanding our DNA, can actually help us to understand Parkinson's. And some of the drugs that are being developed based upon this understanding. And actually, I've used the example of LRRK2. There are drugs now in clinical trials targeting many of the other genes that are involved in genetic forms of Parkinson's, including alpha-synuclein. There's several clinical trials ongoing trying to target alpha-synuclein. Again, we wouldn't be able to do those without understanding the genetics. I put a slide up with all of the people I've worked with. I'm not going to run through these in detail just in the interest of time, but one thing I would really highlight is exactly how collaborative and international all of this types of research is. I work with people in America, over in Europe. We collaborate with people even across in Australia who work on exactly the same in the same area. And of course, we wouldn't be able to do this research without the support of funding bodies like Parkinson'S UK. We're also funded by the Michael J. Fox Foundation and by a variety of government funding bodies like the Medical Research Council. More than happy to talk about answer questions on anything that I've talked about, or more generally about the science of Parkinson's. I may not be able to answer all of the questions, but more than happy to field them. So thank you very much for your attention.
Can I just thank you, Patrick? Can you hear me?
I can indeed, yes.
Can I thank you, because that was really, really interesting. And you actually made it quite simple, so that us non-scientists could understand it. So that's really beneficial. Are there any questions from people? No? I mean, I have a question, if I may. I may have this genetic mutation. So I'm wondering what could happen. You know, would they find a drug for me, or possibly?
It's very difficult to know, and this is something that I'm not a clinician, but talking to clinicians who I work very closely with, many of whom have worked with genetic inherited forms of neurological disorder for many years, is that at the moment, actually, for genetic forms of neurological disease and neurodegenerative diseases, actually, at the moment, we don't have drugs which will make a difference.
Yeah.
We're beginning to see the very first. So if I was giving this talk 5 years ago, actually I'd say, you know, we don't have any drugs for any form of neurological disease in that way. Actually, that's beginning to change. And I mentioned at the end there the company Biogen, who are using a gene therapy approach to try and target the LRT2 gene in Parkinson's. So they're working with a company called Ionis, who are one of the pioneers of gene therapy approaches to brain disorders. And actually, they have now got a drug which is licensed, a gene therapy drug, which is licensed for use in a very, very rare, thankfully very, very rare form of motor neuron disease. And motor neuron disease being the type of disorder that Stephen Hawking had. So the type of motor neuron disease that this drug targets is called spinal muscular atrophy. And actually, it's a form of motor neuron disease that can strike immediately after birth. So it affects infants and very, very young children who are unable to develop motor control and basically die very, very early in life, normally after about a year. And they've used this gene therapy approach to basically stop that disease in its tracks. And children or infants who would have died at the age of 12 months are now living 2 or 3 years and being able to walk and developing motor function that they would never have been able to do. And as I say, fortunately, this is a very, very, very rare disorder. But the principle of being able to target a genetic form of brain disorder and actually stop it, that had never been done before. And so actually we're beginning to see the first suggestions that it might be possible to target these. Now, whether that makes a difference, you know, in the coming years, genuinely I don't know. But it's certainly from the research perspective, I look at that, and that gives me hope that actually there will be therapies coming through. But you know, if we look across at Alzheimer's in particular, you know, the last 10 years has been basically a report after report of drug trials that have gone all the way through to that final stage and not met the clinical endpoints, the changes in symptoms and progression that people were looking for. So the honest answer is, I don't know.
Okay, I'll keep taking my tablets.
That I would certainly recommend. Follow your doctor's instructions, is what I would say.
Is there anyone else who has a question? Since I've
Yes, please, Ruth.
Yes, I do. I have a brother who recently, 2 years ago, died. He had Parkinson's. He's 4 years older than I am, and I've got Parkinson's. We were both probably got it or had it diagnosed around the age of about 70. I'm now 77. Interestingly enough, we think probably that our paternal grandmother had it. So I'm just interested on because everybody says it's only 5%, they reckon, that's genetically handed down.
So it's one of the big challenges with genetics is that geneticists, and I'd emphasize I'm not a geneticist. I work the geneticists provide me with information which I then work on. One of the problems with genetics is that a lot of what they talk about are probabilities, and so it might just be chance that you and your brother develop Parkinson's. It might not be. And actually, if it is the case that you think there is a family history, that might be something that's worth raising with your GP or with your neurologist if you're seeing a neurologist. And I think going back to the last question, one of the challenges with all of this is that actually learning whether or not you have a change in your DNA won't actually make any difference at present to the way in which the drugs which you're prescribed. So in that regard, actually, it doesn't make a difference. But if it is something that you think there is a family history, it is worth talking to your GP or your neurologist about.
Yeah, well, I have actually already done that, and they've taken a blood sample, but there's a lack of funding apparently to process all these things at the moment. What a surprise!
Yeah. One of the things that again is I'm astounded by as time has gone by is how rapidly it's changed, how rapidly genetic testing has become available. So the first sequenced the human genome back in 2000, 2001. It cost something like $200 million to sequence one person's genome. You can now do that in a day and for about £1,000. Wow. And it won't be long before this is something which is essentially routine. Yeah. And that raises a whole host of issues, that sort of non-trivial issues around how do you deal with that information? How do you store it? Should insurance companies be privy to that information? A huge realm of ethical and moral issues that arise from it. But that technology is improving all the time. And so at the moment, funding may well be an issue, but actually it won't be long before it costs £100, and it costs £69 to get an antibody test for COVID-19. It won't be long before it's cheaper than that to have all of your DNA sequenced.
I mean, to my three children, grown-up children, I mean, the twins are in they're 54. To me, it's quite important if they know, or if they can find out there's a likelihood that they're going to develop Parkinson's, because there are things that they can do and drugs that they can take which will help postpone or at least diminish their symptoms from arising.
So one of the things I would one of the things I would recommend if you haven't read this, if you haven't come across it, is Sergey Brin wrote an article actually about 6-7 years ago, I think, in Wired magazine. This is so if you search Sergey Brin Parkinson'S Wired magazine, it will come up. And in it, he talked about the sort of the thought process that he went through in getting a genetic test, but also the way in which he approached that. What does that mean to him? And actually, one of the things he highlighted is that he became very quickly very expert in what people think are the protective things that you could do to maintain as much brain function as possible. So I'd really recommend reading that. But again, you get into a very, very complex area of probability versus genetics versus clinical medicine. And there is a lot out on the internet about this. But I'd recommend the article from Sergey Brin. I read it, and actually I found it very, very interesting to have his perspective on that.
Would you give me the details so we can put it on the web?
I shall try and dig them out now.
Marvellous, thank you. Does anyone else have any questions?
Rose has got a hand up. Rose?
Lucy?
Oh,
Rose?
yes, unmute myself. Yeah, I just wanted to ask you if you could explain the relevance of all this to people who haven't got the inherited version.
Mm-hmm. So this is something that, in terms of the genetics, myself and colleagues at University College are doing a lot of research on at the moment. What I've talked about today is are those rare changes in the DNA, those mutations that can cause Parkinson's. But actually, really where the big change has been in the last few years, probably the last five years, I would say, is understanding much smaller changes in your DNA that are actually present in maybe even most of the population, and that don't cause you to develop Parkinson's, but very slightly increase your risk if you look at enough people. And these are called genome-wide association studies. And basically what you do is you take a lot of people with a particular trait or disorder, and a lot of people who don't have it. And you're talking tens of thousands of people. So the most recent study for Parkinson's took about 40,000 DNA from 40,000 people who had Parkinson's, and about 100,000 people who don't have Parkinson's. And actually they identified changes in LRRK2 that don't cause Parkinson's, but increase your risk. So we think that actually, and this is the hope of the pharmaceutical companies who are not doing this out of the kindness of their hearts, they're looking for drugs that could be used by anybody who has Parkinson's. So their hope is that actually these will, that if they get a drug that targets SERT2 in people who have a mutation, that actually it will also have benefits for the more general Parkinson's population. I've just been sent a message by Simon, by the way, pointing out that we're very close to 8 o'clock and the clap for carers. So Simon, shall I pass back over to you? So I hope that answered your question, Rose.
Right. Can I just say that I think we'll have to have you back in real. Anyway, Simon, sorry, I've interrupted.
I'd be more than happy to.
Say, if there's more questions, we can do it after we clap for the carers. But I know that myself, for one, I always go out on a Thursday night and clap for the carers. So if we want to resume here at 8:05, I've got a question for Patrick that I'd like to ask, if that's possible, after the break. And we can see everyone back here at 8:05. Is that all right, Ruth?
Yeah, fine by me.
Great. Okay. Well, I'll put a little sign up saying we're on a break. So
Back soon.
Intermission.
We'll see each other very soon. Thanks, guys.
See you in a minute.
See you in a minute.
Yeah.
Hi, everyone. Welcome back.
Hello, hello, hello.
I've got a question, but I can wait.
We lost Patrick.
Patrick, well, yes, yeah. Patrick, my question is, do you think there'll ever be a time when a human embryo can be manipulated to remove or alter the cells that Parkinson's inherited cells? Do you think that's possible? I know you talked a little bit about drugs that might be able to do that as well.
So, I mean, technically that's possible now. There are a couple of ways in which that can be done, and actually the sort of the principle of that. If you have an inherited form of disease and you want to prevent that being passed on to the next generation, actually that's been possible for a number of years. And the example that is probably best known, or in terms of a brain disorder, is Huntington's disease. So if you have Huntington's is a purely genetic form of disease. That you only have Huntington's if you have a mutation in the Huntington gene. And what you can do is you can undergo, if you're looking to have children, you can undergo IVF, and the resulting embryos can be screened for the Huntington's mutation. What I didn't go into a detail of when I was going through the genetics bit was that we have 2 copies of every gene. And for Huntington's, and actually this is also true for LRRK2, you only need one of those copies to be mutated to develop Parkinson's or Huntington's. And so what you can do is you can basically screen those embryos, and there's a 50-50 chance that an embryo will have one of the mutations, and you can simply select one which doesn't have that mutation. So that's been possible for many, many years. It's very, very tightly regulated. It's a process called pre-implantation genetic diagnosis. So that's been possible for many years. What is now becoming possible is actually going in and genetically editing that DNA. And actually, this is what we're doing with the worms, with C. elegans. We're using an approach called CRISPR-Cas9, which is basically, it's using a very refined pair of scissors to cut out and stick a different bit of a different letter in. And that allows us to genetically engineer the worms so that they have the mutation that Sergey Brin has. But actually, the principle of that is applicable to any DNA. So we're using it in a worm, but you could use it in a mouse, and it has been used a lot in the mouse, but it can also be used in a human, and it's been used in human cells, and also actually in human embryos, but in a very limited setting. So very tightly regulated, to try and understand developmental, very developmental disorders which are fatal very early in life. There's also reports that actually people have carried out this genetic engineering and allowed babies to be born. Whether that is actually the case, we still actually don't know, but there's certainly there are major ethical issues surrounding the use of this technology. And actually for inherited disease more generally, actually really it's not required. Whether you could do that and specifically treat and change the DNA in cells that are affected later on in life is something which is being actively developed. So I know people are developing this gene editing approach to actually use it, for example, to treat cystic fibrosis, to try and edit out the mutation that causes cystic fibrosis in the lungs of people who develop it, but who are already sort of early adulthood or into adulthood. So the approach is being used. Whether it would need to be used for in embryos, probably actually is not the case. But that technology is out there and it is being used.
Thanks very much.
Are any more questions? I think, Simon, if you think it's okay, if there are no more questions.
Janet wants to ask one.
Oh, there are some, sorry. No, they're out. Marvellous.
Is there any relationship, or is there any serious talk of relationship between Parkinson's and the coronavirus? Because when you hear things like loss of sense of taste and smell, stickiness, and so on, it just makes you wonder. And the flu pan-epidemic in 1918-19 was followed by post-encephalitic Parkinson's. So do you think there's any chance that there might be something similar happening today?
So again, the honest answer is I don't know, and I don't think anybody really knows. Certainly, I work with a neurologist at the Royal London Hospital, a guy called Alistair Noyce, who's done a huge amount of work actually looking at problems with sense of smell very early and actually before any kind of movement symptoms in Parkinson's. And he immediately, when the reports came in of loss of sense of smell and coronavirus, he immediately flagged that up. He's very active on Twitter and he was talking about this a lot and was I don't know whether I would say concerned, but it was something that he was looking at. And I think neurologists, because of the post-encephalitic Parkinson's that you mentioned, are very aware that actually we may have an example in the past where that has been the case. My understanding is that at present, the loss of sense of smell is more to do with actually problems in the nose rather than in the brain. If you like. So it's inflammation in the nose and the initial sensing of smells rather than the way that's processed further into the brain, which I think would probably raise more concerns. But in all honesty, we don't know, and there's a huge amount that isn't known about coronavirus across the board. So I think it's something that is being very actively looked at. And so neurologists trying to understand any neurological symptoms that people who have COVID-19 present with, they are looking at this and are following patients to try and get an idea as to whether there are any consequences down the road that we may not become aware of for a number of years. So it's being looked at. As far as I understand it, I don't think there's any clear evidence that that would be the case, but it is being examined quite closely.
Yes, because there are things like gut problems, and I know if you have a levodopa challenge, you often get a runny nose at some point in that. So there just seem to be quite a few links, and we might actually benefit from all the research that's being done into coronavirus.
That may, and actually, to give you an example of how the different areas of research can benefit across the board. Actually, the people I work with at the Diamond Light Source, back in January, they put all of their other projects on hold and started working only on coronavirus. And they published a structure for one of the coronavirus proteins within about a month, which is incredibly quick for this kind of research. So that's a group who are structural biologists who are working on Parkinson's, but are actually also working on coronavirus. And I think actually you see more and more people learning from other fields. So it's certainly not beyond the realms of possibility that that there could be things that we can't even predict now that may benefit research in Parkinson's from all of this.
Any more questions from anyone? Well, I don't know. I think Ben, I'm going to thank you, Patrick. That was absolutely fascinating, really, really good, and there is no way you can escape from coming back.
Excellent. Well, as I said, I shall look forward.
Very interesting, very, very interesting. I agree with Ruth.
Thank you very much. One thing I would mention.
It was simple. It was the way you put it forward was quite easy for me to follow, which is excellent.
Well, one thing I will say very quickly is I put the link into the Wired interview on the comment. So if you click on that, it goes through, and it's actually quite a lengthy article. So and it was actually from 10 years ago. But thank you very much for inviting me. As I say,
thank you so much.
I will look forward to meeting you all in person when some semblance of normality has returned.
Thank you. Can we give Andrew a clap?
From my point of view, may I say thank you very much indeed. I do not suffer from Parkinson's, so it's a real brand new subject to me, and it's something which I'm so pleased Ruth got you with us today to talk about it. I found it fascinating from my point of view, and I'm sure everybody else as well. Patrick, I do thank you for your time and coming to us to listen to and talk to us and give us an indication of what it's about. Listening to the comments by the members of the branch, I'm sure they found it fascinating. And as Ruth says, we welcome you back anytime.
Lovely, thank you. Thank you very much. Thank you. Clap.
I now have to go and read a bedtime story to my 3-year-old.
Okay, well, bye-bye. Bye-bye. Thank you so much. Thank you. Bye-bye.
Bye.
Bye. Right. Excellent.
Simon, thank you so much again for the technicalities of putting it all together and running us forward there. Do thank you very much indeed. It's been great. It's been really good. It shows we can do this on a regular basis, I hope, I like to think, through your expertise. And again, Simon, thanks so much.
Yeah.
Have we got a raffle?
Thanks everyone. Thanks for coming.
Virtual raffle.
Yeah. Thanks everybody. Keep safe.
Bye.
Thank you.
Bye-bye.
Thank you. Bye very much.
Thank you.